Iron is not a requirement for blood. It is not a universal rule of biology. It is just the specific version your body happens to run on. Almost every animal you can picture, from a bird to a whale to the person sitting next to you, runs on the exact same iron-based system. But right now, somewhere in the deep ocean, there is a creature whose blood is built on a completely different metal, engineered to do the exact same job in a completely different way. Its blood is not red, it runs blue. And by the end of this video, you will understand why human medicine has spent the last 40 years trying to build something remarkably close to it, on purpose, in a laboratory. Every drop of your blood depends on one molecule, hemoglobin, a protein built around a single iron atom that grabs oxygen in your lungs and releases it everywhere else in your body. That design works extraordinarily well, which is exactly why almost every complex animal on Earth was built around it. But it is not the only design that works, and it is not even the strangest one. Somewhere on this planet, an animal exists whose blood chemistry looks almost alien next to yours. Built around a metal most people have never once considered a biological option. And there is one more fact, waiting at the very end of this video. Humanity has already engineered a working answer to the question you're about to ask, and it did not come from science fiction. It came from a real lab, tested on real patients. Cut open a cephalopod, an octopus or a squid, and its blood is not red, it is blue. Not a metaphor, not a filter. An observable fact confirmed in physiology laboratories since at least the 1960s. The reason is a protein called hemocyanin engineered around a pair of copper atoms instead of a single iron atom. When hemocyanin binds oxygen, that copper pair shifts its electron state and produces a deep blue pigment, the exact same functional role that iron plays when it produces red in your own blood. Copper-based oxygen transport looks less efficient than iron at room temperature, but in cold, low oxygen ocean water, it holds on to oxygen with remarkable precision, a design tuned exactly for its environment. Recent structural research on molluscan hemocyanin has revealed oxygen-capturing sites built with more flexibility and surface exposure than scientists expected, a level of molecular engineering still being mapped today. Here is where it gets more useful than you might expect. Horseshoe crabs also carry copper-based blood, and their blood cells react so violently to bacterial contamination that they can detect it at concentrations of parts per trillion. That reaction became the pharmaceutical industry's global safety test, still used right now to confirm that injectable medicines and vaccines are free of dangerous bacteria before they ever reach a human body. Copper-based blood did not turn out to be a lesser design. It became a medical safety standard the entire world quietly depends on, so iron is not universal. Copper proves that on its own, and it is already saving lives in hospitals today. But copper is not even the strangest metal biology has assigned to this exact job. Inside the ocean, there is an animal doing something with metal chemistry that should not be possible. The sea squirt, a soft-bodied filter feeder called a tunicate, concentrates vanadium inside specialized blood cells called Vanadocytes at levels 10 million times higher than the seawater surrounding it. 10 million times. That is one of the most extreme chemical concentration feats ever documented in a living system, first discovered in 1911, and still being investigated more than a century later. Inside those Vanadocytes, the environment is astonishing on its own, an internal acidity roughly as extreme as battery acid, engineered specifically to keep that concentrated vanadium chemically stable and ready. And here is the part that should stop you. After more than a century of research, scientists still do not fully know what that vanadium is for. It is not confirmed to carry oxygen. It is not confirmed to fight infection. Specialized proteins called vanadium-associated proteins have been isolated from these exact cells, and a Vanadocyte-specific marker appears in the body at the precise moment vanadium accumulation begins, evidence of a precisely timed, deliberately built biological system whose true purpose remains, to this day, one of the open questions in modern biochemistry. If you take one fact from this video to repeat today, let it be this. There is a living creature in our own oceans concentrating a metal at levels 10 million times its environment, held in an internal chemistry as extreme as battery acid, and no one on Earth can fully explain why. And if blood chemistry has already been engineered around two metals stranger than iron, the next question answers itself. Has anyone tried building that option for a human body? The answer is yes, and it is not theoretical. Since the late 20th century, medical researchers have engineered hemoglobin-based oxygen carriers, HBOCs for short, blood substitutes built to transport oxygen through a human body without requiring a donor's iron-based red blood cells. Products with names like Hemopure and Polyheme reached large human clinical trials, and some were used in real trauma and emergency medicine, built specifically for situations where donor blood could not arrive in time. The results were not simple. A landmark clinical meta-analysis found that early hemoglobin-based carriers raised the risk of heart attack and death, because free hemoglobin outside a protective blood cell behaves very differently inside the body than researchers first expected. But that setback did not end the field. It redirected the engineering entirely. A newer oxygen carrier called M101, engineered from an unrelated marine worm's blood chemistry, has already been tested successfully in human kidney transplant preservation, keeping donor organs healthier before surgery. This is the promise made at the start of this video, delivered in full. Your blood chemistry is not a fixed biological requirement. It is one working option among several, engineered by nature at least twice already, and human engineers are actively building further alternatives right now, in real laboratories, for real patients who cannot wait, in a field that is still advancing every single year. So the next time someone tells you blood has to be red, tell them about the blue blood inside a cephalopod, engineered around copper and now protecting every vaccine you have ever received. Tell them about the still unexplained vanadium locked inside a sea squirt at 10 million times the concentration of the ocean around it, a mystery no laboratory has fully solved. And tell them about the human-engineered oxygen carriers already tested inside real patients who needed blood and could not wait for a donor. Iron was never the rule written into biology. It is simply the version you happen to be built with. One option among several this planet has already proven can work. If that changed how you think about the blood in your own veins, subscribe for the next one, because the next extraordinary design, hiding in plain sight right under everyone's assumptions, is already lined up and waiting, and it is stranger than this one.